Okay, moving on to genetics practice 2, beyond the basics. This has a lot of things that are considered non-Mendelian genetics, meaning that these don't necessarily work the same as simple dominant recessive. Sometimes there's something different that's going on here. Maybe there's more than 2 alleles. Maybe there is some sort of phenotype that's neither dominant nor recessive. Let's look at this first problem. This one is incomplete dominance. So, in radishes, the gene that controls color exhibits incomplete dominance. All right. Pure breeding, this word pure breeding is an important one to know. All it means is homozygous. Homozygous. The reason I know it's homozygous is because pure breeding means whenever in this case, red radish, is crossed with another red radish, you always get a red radish, meaning that it is homozygous, right? So, pure breeding red radishes crossed with pure breeding white radishes make purple radishes. So that's a third phenotype. So, if I was to look at this out here in cross form, I'm going to do a little tiny cross over here to show what they're trying to tell you. So, and I'm going to use 2 different alleles, big alleles. I normally wouldn't suggest use different letters except for scenarios like incomplete dominance. But we're going to use, let's say, R and white, W for white, right? So R would be red, W would represent the white allele, right? So pure breeding reds crossed with pure breeding whites, right? Remember that pure breeding means homozygous, yields purple radishes, which means purple radishes have 1 allele, 1 red allele, and 1 white allele. So I think we can do this cross now that we kind of understand a little bit about how this works. First things first, as usual, let's make a key. I have started this key up here with the big R meaning red, big W meaning white. Now, it might help you when you make a key for something when it comes to incomplete dominance to make a key for the genotypes as well. In this case, I'm going to put big R gives us red, big RW gives us purple, and WW gives us white. Right, so this kind of tells us, since we have this different phenotype in here of purple, this kind of helps guide us and let us know what we need to put on each side, because it's not as simple as just homozygous, heterozygous. There's different phenotypes in here. Okay, step 2. Parent genotypes. Parent genotypes. Okay. What are the genotypic and phenotypic ratios? So we're looking for ratios. Remember that's like colon, like 1 for 1. When you cross a purple radish with a white radish. So we know a purple radish is big RW, and we're crossing that with a white radish, which is WW. Okay, parent genotypes. Sorry, that's my dog. Let's put 1 parent on top, big R, big W, and let's put 1 parent on the other side, WW, and you end up with RW, WW, RW, and WW. Okay, what are the genotypic and phenotypic ratios? Well, if we're looking at a genotypic ratio, I'll look at that first, and we only get 2 total genotypes out of this, and that is RW and WW. And those are in a 1-to-1 ratio, meaning for every RW, there is a WW. This is kind of like a 50%. You can think of it like counting 2 to 2 and reducing it to 1 for 1, right? That's our ratio of RW to WW. Now let's look at phenotypic ratios. Purple. and white are all we get out of this matching. So I'm going to put purple first, and then I'll put white. And also we get the same ratios here, a 1-to-1 ratio of purple to white. So that's how you do a pretty simple cross for this one. I forgot to put my little step 3 down here, but we did that cross, right? Okay, then move on to the next one. Certain breeds of cattle show incomplete dominance in coat color, right? So when pure breeding, here's that word again, pure breeding. I'm just going to type homo up here, homozygous. Okay, red cows, so homozygous red, are bred with pure breeding. White cows, right? Homozygous, white, red. This should look somewhat familiar using red and white. The offsprings are considered roan, which is that third phenotype, which is a pinkish coat color. Not super important, but it's interesting to note that there are 3 phenotypes here. Summarize the genotypes and phenotypes of the possible offspring when a roan is mated with a roan. Okay. Step 1. Make a key. Okay. Pure breeding red cows. I mean, I don't see a reason not to just use the same letters I just used. R is going to be red. W is going to be white. Right? Now, based on what we saw up here in that initial cross, I'll go ahead and show you. Pure breeding red, it's the same exact thing. Pure breeding reds crossed with pure breeding whites yields a roan in this case. So RW would be roan. So R is red, W is white, and I'm just gonna, just since this is an incomplete dominance one, I'm gonna make a little key out here with big R, RW, and WW, right? So we know big R is red, WW is white, and RW is roan. Okay, we have a good key made here. Now, parent genotypes. Okay, parent genotypes, what are they? Genotypes and phenotypes of the possible offspring when a roan cow is mated with a roan bull. So that means that both of them are RW. So let's just put RW times RW here. Okay. Third step, cross. All right, I'm going to put RW on top, RW on bottom, big R. This part should be pretty familiar to you if you finish the other worksheet 1. Okay, we've got a red, we've got a white, and we've got 2 roans. Okay, so we can summarize the genotypes and phenotypes however you want. I tend to look at these and just think of a percentage here. So let's do red. What percentage of these cows are red? Well, 1 out of 4 is going to give you 25%, right? So saying 1 out of 4. You can put either one of those. Let's look at roan. Roan is right here and right here. So that's 2 out of 4. 2 out of 4 or 50%. And then finally white, much like red, is also going to be 25%. So those are our phenotypes, right? But let's look at genotypes here. Not much different in this case, right? Big R is going to give you red, so it's also going to be 25%. Big R, big W is also going to give you 50%, just like roan over here. Oop, let me make that 5 better. Oop. Okay. 50%, and then WW is going to give you 25% because there's only 1 out of 4 of those. And that's really all there is to it. That's all there is to it. Okay, let's move on to another one here. Codominance. Codominance is when 2 alleles are fully expressed, and you'll see what I mean here in a second. This is blood typing. So the way to do this one, and I recommend using the alleles that I think you should use. Step 1 is going to be make a key. All right, step 1, make a key. Okay, I'm going to use and this is kind of a key that you just kind of gotta trust me on. I'm going to use IA, type A, use IB, type B, and I'm going to use little I for type O. Type O blood. All right. So you'll notice there's big I's, those are dominant, little I is recessive. These big I's are codominant with each other, meaning that they are both fully expressed if you have that allele. So this is part of our key, but we also should probably make some sort of genotype key. So let's see what possible genotypes. Remember, this is different because we have 3 alleles. 3 alleles instead of 2, right? So to get type A blood, you can have big IA, or you can have big IA and then the little I. Those will both give you type A. Let's do type B. Big IB, or big IB, little I. Okay, let's do type AB. Type AB blood. If you have type AB blood, you have big IA, big IB. And if you have type O, you have little I, little I. Now that's a very in-depth key, but we kind of need it for this one when you have 3 alleles because there's more possibilities. And I'm actually going to move these over. Give me 1 second here. So I got some more room. So I got a little bit more room over here to write stuff. Because that was a big key. I'm just going to put a slash. IAI, IBI. Okay. Remember, 3 alleles leads to much. These are again phenotypes: type A, type B, type AB, type O. Lead to kind of more phenotypes, right? So a man with type AB blood marries a woman with type B blood. Her mother has type O blood. List the expected phenotype and genotype frequencies of their children. Okay, we've made a key. Step 2, parent genotypes. Parent genotypes. Again, I'm kind of realizing I don't have quite enough space. I'm just going to put GTDs over here. Okay. First parent is type AB. So I'm going to put type IA and type IB. Crossed with the second parent is a woman with type B blood. Okay, now let's look at type B. Now this is a bit of a problem here because we have 2 ways to get type B, right? We have IB, or we have IB little I, right? Now how do we know which one to put? It has this other clause here which says her mother has type O. Now type O, if you look at our key, is little I, little I. If her mother had type O blood, what allele did her mother have to give? If you need to pause and think about it, you can. Well, if her mother had type O blood, that means mother had to give daughter a recessive I allele, right? Had to, because she didn't have a big I to give. So we know that her daughter has to have, if she has type B blood, she has to have a little I next to that. Okay, now we have the parent genotypes. Now we can cross. Let's cross. Okay, IA, IB, and then on this side we'll put IB little I. Okay, build them out here. IA, IB, 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 IAI, and IBI. Okay. Now what we need to do is figure out the phenotype and genotype frequencies, and we can use our key to help us here. And I'm just going to put in the top corner what blood type it is. A and B gives us AB. Okay, let's look at this one over here. IB, look at your key. That's going to be type B. IAI is going to give you type A. And then finally, type IBI is going to give you type B. Okay. Now we just need to come up with phenotype and genotype frequencies. Well, that's easy. Our phenotype is what the blood type is, and our genotype is the actual letters. So let's look at the actual letters here. We have IA, IB. What's the frequency of IA, IB? Well, there's 1 out of 4, so that's going to be 25%, or in frequency form, 0.25, right? All right, let's look at the second one here, and I'll just to save me some room here. IB is equal to also 1 out of 4. There's only one IB out of 4 total is going to give you 0.25 as well. All right, let's move down here to IAI. That's another one we have. IA little I is also going to be 0.25. And then finally, IB little I is also going to be 0.25. So 25% of each of these different genotypes. Now, genotypes are up here. Phenotypes we're going to do below. Phenotypes are going to be on the second line down here. Okay. First phenotype we have, type AB. Type AB blood. Now, 1 out of 4 is type AB, meaning the frequency of type AB blood is 0.25, 25%, however you want to look at it. All right. What other blood type do we have here? Well, we have type B as a possibility. Type B. How many type Bs do we have? 1, 2 out of 4. So that's actually going to be 2 out of 4, or 50%, or 0.5 as far as frequency. Okay, our final type here is going to be type A. And of course, there's only 1 out of 4 of those, which is going to give you 0.25. Okay, those are kind of some of the scenarios that you might see. That is, again, that's non-Mendelian genetics. So it's stuff that doesn't quite conform to the simple dominant recessive that we were format that we were doing before. Okay, this is the first half of the second genetics worksheet. My next video will be over the other side of this same worksheet. Thank you for listening.